Moving along to concept 26.4, it says an organism's evolutionary history is documented in its genome. Of course, prior to the modern science that we have today, we had no way of looking at the genome of these creatures, either present or past extinct creatures, or any way of knowing, except by looking at morphology, how all of these creatures fit into certain categories. So it's only been in the last 50, 40, 30 years that we've had the ability to do so much of what we can do now that as it relates to this idea of evolution and change over time. So the molecular approach allows us to understand phylogenetic relationships that can't be determined by non-molecular methods such as comparative anatomy. So particularly if we're comparing, say, if you want to know how a butterfly and an octopus are related, well, how are you going to compare their anatomy to understand that? You wouldn't be able to do that. So if you're trying to compare very divergent species that obviously diverged quite some time ago and they have very little in common anatomically, you can't really do that except through a molecular fashion. So molecular biology has helped to extend this idea of determining who is related to who and how to evolutionary relationships that are far above and below the species level. We can look at very widely flung creatures. We can compare a human to an oak tree and find out where the relationships are and where they have things in common, and perhaps when their the last common ancestor was, things obviously you couldn't do just by looking at anatomy. So let's look at the genome. Comparing nucleic acids or other molecules to infer relatedness is a valuable approach for tracing organisms' evolutionary history. And we have found that different genes evolve at different rates, even in the same evolutionary lineage. For example, DNA that codes for rRNA changes relatively slowly and is useful for investigating branching points hundreds of millions of years ago. The reason that the DNA that codes for ribosomal RNA changes so slowly is because it's so important. So any slight variation or mutation is likely to be detrimental. So you have a conservation of that DNA, of that strand of the DNA. So that DNA that codes for the ribosomal RNA has been around for a long, long time, relatively unchanged. And so that allows us to investigate species where the branching points occurred a long, long time ago, hundreds of millions of years ago. And you can reflect that, or you can put that with, say, mtDNA, which evolves quite rapidly and can be used to explore recent evolutionary events. mtDNA is the mitochondrial DNA. If you'll remember, the mitochondria have their own separate DNA. That organelle within the cell has its own separate DNA, and that evolves fairly rapidly. So we can use that sort of as a method for examining recent evolutionary events. We know from our previous chapters that one of the ways that change occurs over time is through gene duplication. We have oftentimes a particular type of error occur in the processing where you have gene duplication, and that increases the number of genes in the genome. And of course, when you have an increased number of genes, you have an increased opportunity for evolutionary change. Repeated gene duplications results in something that we call a gene family, basically a group of related genes within an organism's genome. So oftentimes you'll have these duplication events occur multiple times, and these duplicated genes become like a family. They are obviously related in a way, if you want to talk about genes being related to one another. Like homologous genes, duplicated genes can be traced to a common ancestor. One variety of genes that we can look at are called the orthologous genes. Let's try that again. Orthologous, like homologous. Orthologous genes are found in a single copy in the genome and are homologous between species. Man, that's like a tongue twister. Orthologous genes are homologous between species, and they can diverge only after speciation occurs. So let me show you a picture to kind of clarify what I'm talking about here. So an orthologous gene would be basically one that gets passed down from the ancestral species to the species that diverged from it, and then there's likely to be a few changes. So here you have the original gene, And then here you have two different species, species A and B, and these are orthologous genes within these two species. You'll find that they're very similar. You can see the little red mark and the yellow mark. That's where some minor changes have taken place, but you could trace back species B and species A. First, you can say they are somewhat related because they have these orthologous genes, and you can trace both of them back to this ancestral species with that ancestral gene. So similar to tracing characteristics back, like saying how the crocodiles and the birds both brood their egg nests, you might trace that back to its common ancestor and say, well, then perhaps the dinosaurs did too. So that's doing the same thing, but we're doing it at a molecular level, at a genetic level with these orthologous genes. The other type of gene we can look at is called a paralogous gene, and a paralogous or paralogous genes, they result from gene duplication and are found in more than one copy. There are more than one copy of them in the genome. They can diverge within the clade that carries them and often involve new functions. So again, let's look at a picture so we can understand what we're talking about. Here we show the ancestral gene in the original species C, and then we have species C here after many generations, but you can see that something has happened to that particular gene we're looking at. Not only has it duplicated, but it has diverged. So we're still talking about the same species here, but within that species we're starting to get some genetic divergence to where this gene perhaps had one purpose, and now there's two copies of it in this this descendant, and there are two different varieties. And sometimes what can happen is perhaps this gene had a particular job, and now in the descendant there are two copies, and maybe one of them still does the same job as this one, but perhaps the second one takes on a new job. So because you have duplication of genes, you're creating more genetic material, and then that gives the possibility of different mutations and different possibilities. So now perhaps this descendant has this gene, and maybe it still does the same job as the original, but it has this second gene because of the duplication error, that perhaps it goes on and undergoes its own mutations and begins to do something completely novel and completely new. So here are the two different cases of looking at molecular data, genetic data, the same way we were looking at traits earlier when we were talking about the dinosaurs and the birds, or the same way you might look at homologous structures like the bones of the forearm between the bat and the human. Here we're doing the same thing, but we're doing it at a genetic or molecular level. So you have the orthologous genes where you have speciation with divergence of the gene, and so the genes are not quite the same, and this is after they have split into two species, but you can still tell that they are related to one another and that they came from this common ancestor. And then you have this particular type where you have the same species, but there's some duplication events, and the genes themselves are beginning to differentiate from each other, and that may mean that now there are new possibilities, but you can still tell that this is a duplicate and that it came from this ancestral gene to begin with. These orthologous genes would be very equivalent at the genetic level to the homologous structures that we talked about earlier. So these orthologous genes are widespread and extend across many widely varied species. The homologous structures you're only going to see likely within groups of creatures that are fairly closely related. Like you're going to find the forearm bones of almost all mammals are very similar. You know, the whale, the bat, the cat, and the human. But you wouldn't expect to find that in, say, a butterfly or a fish. These orthologous genes, however, can be widespread across a huge swath of living creatures. For example, humans and mice diverged about 65 million years ago, and 99% of our genes are orthologous. They show common ancestry. And now, I don't want you to think that that means that I'm saying that 99% of our genes are the same as mice. They're not, but they are orthologous. In other words, that doesn't mean they're alike, but they have a common ancestor. The genes themselves have a common ancestor. Now, since that divergence of species, they have evolved and changed quite a bit. So now our entire genome is not as similar to the mouse as perhaps it once was back further in time. As a matter of fact, you can even go crazier than that. 50% of the human genes are orthologous to those of yeast the tiny little creature that gets put into bread to make bread rise, or that makes alcohol in the fermentation process. And that has been over 1 billion years of divergent evolution, and yet we can still see the relationship between our genome and the genome of yeast, a very simplistic single-celled creature. One more thing to keep in mind when you're looking at the genome of these creatures and trying to infer relationships is that the gene number and the complexity of the organism are not strongly linked. So you might think that, well, the more complex the creature is, the larger its karyotype, the more chromosomes it's going to have and the more genes it's going to have. Well, that's not really the case. When we talked about the chromosomes, we found that there was really no relationship, and we just want to hit that again. For example, humans have only 4 times as many genes as yeast. And a yeast is a single-celled eukaryote. I mean, we are vastly more complex than yeast, but we only have about 4 more times as many genes. So there's no direct relationship between the number of genes and the complexity of the creature. It turns out, it seems that what causes or what allows for the complexity is not the number of genes, but the versatility of the genes. In humans, or in a lot of the, perhaps I guess you could call them more advanced, more complex creatures, a single gene can encode multiple proteins that can do multiple jobs depending on how you turn it on or turn it off and how it's affected by some of the other genes we've discussed. So a single human gene can potentially create multiple proteins that perform different tasks in a variety of tissues, whereas a single gene in the yeast perhaps only has a single job to do, and it only does that one job. But it seems like the more complex the creature, not necessarily the more genes, but the more versatile those genes are. We're going to take a break. We'll come right back with concept 26.5, which is how do we put all this on a time scale? When we say this dinosaur lived X number of years ago, what's the molecular or genetic basis for saying that? We're going to talk about molecular clocks in just a moment.